Hi everyone, today we are going to talk about prokaryotic organisms. Prokaryotes, you remember those. Unicellular, simple organisms, no nucleus, right? No nucleus. They don't have as many of the complex cell organelles that our cells do as eukaryotic organisms, right? And when you think of a prokaryotic organism, what's an example of one? What comes to mind? Yeah, bacteria, right? That's the one that I think of. There are thousands of species of bacteria. That's the most common example, I would say, of a prokaryotic organism. So this week in lab, virtual lab, I know you can't see me, but I'm doing the air quotes, you're going to be looking at bacterial transformation. So it's important to have an understanding of kind of how these bacterial organisms, their structure, their function, how they reproduce. We're going to talk about bacterial transformation, what it is and how it works today, so that you have some background knowledge, kind of laying the groundwork for the lab you're going to do on Wednesday. So prokaryotic organisms are very diverse. They've been here a long time. Do you remember from the evolution unit that prokaryotic organisms are some of the first fossils we found on the earth, right? They are very diverse. They're able to live in all different kinds of places, function under all different kinds of environments. They are mostly unicellular. They do come in a variety of shapes. They're well organized and they're able to perform many life functions. So some of the shapes that are listed here at the bottom, spheres, rods, and spirals. So that looks like this. This is the spherical or the coccus-shaped bacteria. This is actually a picture of Staphylococcus aureus, which is the bacteria that is involved in a staph infection, or MRSA, if you ever heard of something like that. Streptococcus bacteria, they're not the ones in this picture, but they have the same shape. If you've ever had strep throat and the doctor did a strep culture where they stuck that thing down inside of your throat and gag you, right? And then they pull out the cells and look at them, they would be in this shape because that's the shape of the bacteria that's doing the infection. You have rod-shaped bacteria, and then you have these spiral ones. So bacteria are able to reproduce very quickly. They have a very short generation time. It says on the slide there that they can reproduce every 1 to 3 hours, which is very fast. They reproduce exponentially, right? If you've ever seen a graph of that, it's like 1 divides into 2, divides into 4, and so on and so forth, right? So they divide very quickly. Many prokaryotic organisms are able to do this, quick division in favorable environments, by this process called binary fission. Binary fission is essentially just mitosis for bacteria. Single cell divides into 2, 4, 8, 16, and so on, right? And some of the keys to this ability to reproduce so quickly is that they are very small bacteria, right? You can't see them because they're microscopic. They're very small. They undergo mitosis, so that's a quick process, and they have these really short generation times. So when we're talking about binary fission, how they reproduce, we're going to examine 3 factors that give rise to the high levels of genetic variety that can occur in bacteria. What did I just say? Genetic variety, genetic diversity. I just said they do mitosis, right? Well, not all bacteria are exactly the same, right? There are things or mechanisms that incorporate genetic variety into bacterial species. That's how we've gotten so many of them. So we're going to go through and we're going to talk about some of those ways. We know, of course, that they reproduce very quickly. There is the chance for mutation, so that's going to introduce that genetic recombination. So let's look at those things here. Binary fission first. Binary fission is the process that yields identical cells. So this is like mitosis, you can think of it, right? The parent cell splitting into the 2 daughter cells. Very low chance of mutation there. It could happen, but what helps to increase that genetic variety or assist in that genetic variety is that if a mutation does occur during this process, because bacteria reproduce so quickly, those mutations accumulate very fast in a population. So it could be one mutation. Now imagine exponential growth, right? Going like this, up on a graph, right? That's going to take that one mutation and it's going to just accumulate very, very fast. And that can increase genetic variety in a population of prokaryotic organisms like bacteria. So new mutations are a major source of variation, but there are some other mechanisms that can bring together prokaryotic DNA from different individuals. So bacteria are able to do this thing called horizontal gene transfer, and we're going to look at what that's all about. So transformation and transduction. The first one, transformation. The genotype and phenotype are altered by the uptake of foreign DNA from its surroundings. Whew, that's kind of a mouthful. Transduction is when the DNA is transferred between prokaryotic organisms by viruses that carry prokaryotic DNA. So I'm going to back up for a second. Transformation is when a bacterial cell is able to take in a piece of DNA from its surroundings. And that's what we're going to do in our virtual lab this week. You're going to be giving the bacteria resistance to an antibiotic through a piece of DNA that they would take in from their environment, incorporate it into their genome, and then express that trait. Transduction is done by the insertion of genetic material by those bacteriophages. They're those things that had the heads, right, and then the body and the tail fibers, if you remember those from our viruses last week. So that's two ways that you can sort of share this genetic information between bacterial cells. And the third one that we're going to talk about is conjugation. In conjugation, DNA is transferred between two prokaryotic cells that are temporarily joined together. This is always a one-way process where one cell donates to the other one receives it through this thing called a sex pilus. Very X-rated photo of bacteria having sex here on the screen, right? Haha. But the one will extend, they call these fimbriae or fimbri, these pieces going around. They kind of look like cilia if you're thinking about your cell organelles. Similar idea. One of them extends and becomes a sex pilus, and the DNA contained within this donor cell is transferred to the recipient cell. And it attaches, and then it transfers that genetic material. So you might not have known that before, that bacteria are actually able to transfer DNA to one another. So the ability for bacteria to do this and form that sex pilus results from a particular piece of DNA called the F factor. The F in F factor stands for fertility, so the ability to pass on that DNA, just like if you had a fertile offspring or fertile organism that was passing on DNA to its offspring. So it can be part of the bacterial's DNA, or it can be a plasmid that is inserted into the DNA. So the word plasmid is essentially just a piece of DNA that is foreign to the bacterial cell. So plasmid is DNA that the bacterial cell is going to take into its genome, whether it gets it via conjugation like this or transformation. That's what a plasmid is, just a piece of DNA that's being incorporated into the bacterial's genome. So cells without the F factor are the recipients during the conjugation process. So here is the recipient cell, here is the donor cell. Bacterial chromosomes are circular. They're not like ours. They're not segments or, the spiral staircase DNA. They're actually shaped in this circular shape. So that's what it looks like. Here's our bacterial chromosome, and here is our plasmid. Remember, a plasmid is a foreign piece of DNA. So this is an F plasmid. This is a plasmid that codes or contains the genetic sequence that codes for the proteins that build this mating bridge or sex pilus between the two cells. So the mating bridge forms, and the one strand of the plasmid breaks and enters the F negative or the recipient cell. DNA replication will occur in both cells, and now the recipient cell has the ability to form a sex pilus. So it will pass on that trait to another bacterium now that it has the ability to express the proteins to build this mating bridge. And that's what this is all about. So now you have the original cell, and then you have your recipient cell that has both its own DNA, and now it has incorporated the plasmid, or the ability to pass on that trait of making a sex pilus to share genetic information. So the F factor can also be incorporated into the chromosome, and it becomes kind of like how HIV gets inserted into our DNA, right? In our genome, similar idea here. It can be inserted into the bacterial's DNA. So in the lab this week, you're going to be working with an R plasmid, and the R stands for resistance. Some bacteria have resistance genes, which code for enzymes that destroy antibiotics. Antibiotics, right, are what you take when you have a bacterial infection. It's going to kill the bacteria. It kills them by essentially destroying their cell walls, and then they get in and destroy their DNA. So these genes are carried on pieces of foreign DNA called R plasmids. Natural selection will favor, of course, bacteria that have these R plasmids, so then you get more and more resistant bacteria. We talked about that when we did evolution, and we talked about the number of MRSA infections that have gone up over the years because the bacteria that infect us are becoming resistant to the antibiotics we take, right? That's why you should try to limit your antibiotics if you can throughout your lifetime. And we talked about that as well earlier in the year. All right, so that is the end of our talk on how bacteria exchange genetic information. I want to show you a quick segment of a video here on those different processes. It's a little cartoony, but sometimes I think things that are a little cartoony are the easiest to understand. She's going to start with bacterial transformation, which is, and she's going to actually show you a cartoon of the procedure you're going to do in lab this week.
Transformation occurs naturally in some species of bacteria, but it can also be induced in the lab. In order for transformation to occur, competency must be induced. For transformation to happen, bacteria must be in a state of competence, which might occur as a time-limited response to environmental conditions such as electrical charge, temperature, starvation, or cell density. Some bacteria are able to take up plasmids or other fragments of genetic material from their surroundings under certain conditions. Free-floating DNA can then be picked up by competent cells. The exogenous DNA is incorporated into the host cell's chromosome via recombination. Transformation can also be induced in a lab. There are several methods used in laboratories to induce competency. One of the most popular and easiest ways to induce competency in bacteria is the heat shock method. Heat shocking involves exposing bacteria to a moderately high temperature for a brief amount of time. Immediately following the heat exposure, the cell culture is then put on ice. This sudden extreme temperature change causes specialized surface proteins on the cell membrane to
So if we were in school, I would have had my hot water bath on the demo desk, and you would have done this with the bacteria. The bacteria we would have worked with in school would be E. coli. You would put the E. coli and the plasmid into the water bath, and what this actually does is it scrambles up the phospholipids of the cell membrane in these bacterial cells. So we talked about how temperature has an impact on cell membranes, right? When the temperature goes up, they become more fluid. When the temperature goes down, they become more densely packed. So by heat shocking them, you're opening up those cell membranes. The plasmid is able to enter the bacterial cell, and then by immediately putting them on ice, you're causing the phospholipids to become closely packed very quickly, and that's going to seal in that plasmid, so now it is inside of the bacterial cell.
To allow the plasmid to pass through, bacteria are able to internalize the plasmids, and they can then express the genes on those plasmids in the form of proteins. Another form of bacterial genetic recombination is transduction. Transduction is the injection of foreign DNA by a bacteriophage virus into a host bacterium. Bacteriophages can accidentally transfer genetic material from one bacterium to another. In transduction, DNA is transferred from one bacterium to another by a virus or viral vector. Transduction can also be used in the lab to introduce a foreign gene into a bacterial host cell's genome. Transduction happens through either the lytic cycle or the lysogenic cycle. When bacteriophages normally infect a bacterial cell, the virus's genetic material harnesses the host cell's replicational, transcriptional, and translational machinery to reproduce itself. When the new bacteria are formed, the new bacteriophage
So just like viruses can infect our cells by injecting their genetic material, they can do the same thing to bacterial cells. And I want you to take a second to look at this picture. Do you see how small the virus is in comparison to the bacterial cell? That's why, right now in this pandemic that we're in, and they're encouraging everybody to wear masks, it's important that your mask be of a certain material to block this virus, not chances are the things that we're using, like, I've made the cloth masks and I've used cotton fabric and I've double-sided it. That's not fully effective at blocking this virus that is, of course, the coronavirus doesn't look like this, right? We talked about how it's the circle with the spikes on it. But just for reference, that a virus is very, very small, much smaller than a bacterial cell as it's able to infect it.
Must package their DNA into their new capsids. This process is not perfect. We find that sometimes small pieces of bacterial DNA will get packaged along with the bacteriophage's genome. When the new bacteriophages infect a new host cell, the bacterial DNA will be injected along with the viral genome. Another way that genetic recombination in bacteria can occur is conjugation. Conjugation is the transfer of genetic material between two bacterial cells in direct contact with each other through a cytoplasmic bridge. In bacterial conjugation, bacterial cells are able to exchange their genetic material. Remember, this is not done as part of a reproductive process. Conjugation is made possible due to the presence of an F plasmid. The F plasmid is a fertility plasmid. The F plasmid contains genes necessary that allows the bacteria to form a sex pilus. The sex pilus can penetrate the cell wall and membranes of another bacterium. The sex pilus forms a cytoplasmic bridge with another bacterium. Once that cytoplasmic bridge forms, the F plasmid is then replicated and the copy of the plasmid will be transferred to the other bacterium. When the bacteria receives its copy of the F plasmid, it then has the ability to now create its own sex pilus and can go give copies of its F plasmid to another bacterium.
All right, you get the idea. So bacteria reproduce essentially through binary fission, where they would be making exact copies of themselves. But there are these three different methods of incorporating DNA from either another bacterial cell, a virus, so that's going to be incorporating new sequences of genes into their genome so that they can express traits that they might not have been, quote, born with. All right, so this week we're going to look at bacterial transformation in that lab that we're going to do. That lab famously is usually on the AP exam. I have no idea this year if it's going to show up in one of the free response questions, but who knows. So we're going to go through that this week, and now you have a little background on these different processes for getting that genetic variety into bacteria. Thank you, everybody.